It seemed a far-fetched idea. Could the DNA from a Queensland sea sponge shed light on one of the biggest mysteries in animal genomes?
I was in a lab which was working in the sponge, so I guess that's where that idea came from.
Associate Professor Emily Wong and her colleagues were determined to better understand a certain part of DNA sequences called enhancers.
They are regions in the genome which control when and where genes are expressed. We think around 30% of the genome has some sort of annotated enhancer function.
Enhancers perform a critical role in influencing cell identity and certain traits such as size, coloration, and disease susceptibility in multicellular organisms. While they take up a huge proportion of the genome, they're particularly hard to find.
A lot of these sequences, they change really rapidly, so they evolve really rapidly. So the methods that we use to identify protein coding genes across genomes don't work well with enhancer sequences.
That means they're still poorly understood. In fact, they could be considered the dark matter of the genome. This may be why scientists hadn't found any enhancers that were similar across the animal kingdom until the sea sponge experiment.
When we first saw the results, I was very skeptical. I didn't believe it.
Associate Professor Wong and her collaborators at the University of Queensland extracted enhancer DNA from the sea sponge and injected it into a single cell from the embryo of a zebrafish. While the enhancer sequences found in the sponge were very different to those in the zebrafish, they still successfully drove the expression of a protein in zebrafish cells. The findings, published in Science, suggest humans, mice, zebrafish, and most likely the entire animal kingdom share enhancer regions that drive the expression of certain common genes. And given sponges and humans last shared an ancestor more than 700 million years ago, it means the functional mechanism has been preserved throughout evolution. Associate Professor Wong says this discovery means there's still a lot more to uncover about enhancers.
We really need to focus on understanding these regulatory elements and understand how they function.
That's the mission behind her research at the Victor Chang Cardiac Research Institute in Sydney. Solving the puzzle will help scientists better understand basic biology and genome evolution, as well as predict diseases.
We are looking especially in congenital heart disease. We're trying to understand how genetic variants at enhancers could impact gene expression and that could lead to a disease.
Associate Professor Wong says she feels incredibly honoured that her work has been recognised with the prestigious Fenner Medal from the Australian Academy of Science.